Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the key quorum sensing systems in Pseudomonas aeruginosa, from how it works, how they interact with one another, and their effects at high cell densities. Now, during infection, Pseudomonas aeruginosa utilizes three quorum sensing systems to help regulate things like virulence factor production, biofilm formation, and the overall colonization of a host. Essentially, these quorum sensing systems work together and allow bacterial cells to assess their population density, adjusting their behaviour accordingly. Now, the three key quorum sensing systems in Pseudomonas aeruginosa are the LasR system, the RhlI/RhlR system, and the Pseudomonas quinolone signal or PqsR system. Now together, these systems enable Pseudomonas aeruginosa to detect and respond to its own population, functioning as a mechanism for intra-species communication. Now there is a fourth system known as the autoinducer 2 system, and this is based on the Lux quorum sensing system seen in other bacterial species. And this serves as a universal signalling system, allowing Pseudomonas aeruginosa to sense the presence of other bacterial species, functioning as a mechanism for interspecies communication. Now together, these systems help Pseudomonas aeruginosa navigate the complex microbial environment and potentially coordinate behaviours with or compete against other microorganisms. And for the rest of this video, we are going to dive a little deeper into the key quorum sensing systems in Pseudomonas aeruginosa, starting with the LasR system. Now the genes required for this quorum sensing system are LasI and LasR, which are encoded separately with their own promoters and do not form part of an operon, which we can see here. And so under low cell density conditions, these genes are transcribed and translated independently. The LasI gene encodes for the LasI enzyme, which is responsible for synthesizing a specific N-acyl homoserine lactone autoinducer, which in this instance is 3-oxo dodecanoyl homoserine lactone, commonly abbreviated to 3-O-C12-HSL. Now this autoinducer has a homoserine lactone core with a long 12-carbon hydrophobic tail, which has a ketone group located at the third carbon, and all of this links into its function. You see, the long hydrocarbon tail allows this autoinducer to diffuse freely out of the cell and into the extracellular environment, where it begins to accumulate as cell density increases, setting the stage for activation of the Las quorum sensing system. Now at low cell densities, the environmental concentrations of our 3-O-H12 homoserine lactone remains relatively low. However, as cell density increases, so does the accumulation of our autoinducer molecule. Now at high cell densities, the autoinducer will have accumulated in the external environment, and due to its composition, just as it diffused freely out of the cell, it can freely diffuse back into it. And this is where our cytoplasmic concentrations start to increase. Now whilst this is happening, the transcriptional regulator for this quorum sensing system, which is LasR, has been transcribed and translated. And once a minimum activation threshold is met, our 3-O-C12 homoserine lactone autoinducer will begin to interact with the LasR protein, and this interaction triggers LasR activation. And essentially this is where there is a conformational rearrangement of the LasR protein, exposing a dimerization interface, and this allows two activated LasR proteins to bind together, forming a stable homodimer. Now once this homodimer forms, it can interact with specific DNA sequences due to an altered binding affinity, allowing it to turn certain genes on or off. Now one of its primary targets is the LasI promoter, leading to increased transcription of the LasI protein, further amplifying the production of the 3-O-C12 homoserine lactone autoinducer via a positive feedback loop. And what this does is it ensures that once a quorum of bacterial cells is reached, the response is rapidly intensified. Now beyond regulating its own system, the activated LasR protein also controls the expression of multiple virulence associated genes and biofilm associated components. And in addition to all of this, the LasR system has what we call hierarchical control over both the RhlR and PqsR quorum sensing systems. This essentially means the activated LasR protein directly upregulates components of these other quorum sensing systems, helping them to activate, but only after the LasR system has been induced. And what this does is it creates a temporal cascade of gene expression, allowing the cell to optimize resource allocation and virulence expression. Now, the RhlR quorum sensing system is very similar in structure and function to that of the LasR quorum sensing system. It has two genes, RhlR and RhlI, and under low cell density conditions, these genes are transcribed and translated independently. And just as we saw earlier, the RhlI gene encodes for the RhlI enzyme, which is responsible for synthesizing a specific N-acyl homoserine lactone autoinducer, which in this instance is N-butyryl homoserine lactone, commonly abbreviated to C4HSL. Now, this autoinducer has a homoserine lactone core, and coming out of this is a short 4-carbon hydrophobic tail, and all of this links into its function. The short hydrocarbon tail allows this autoinducer to diffuse freely out of the cell and into the extracellular environment, more so than our previous autoinducer of the LasR system. Now, once it's outside the cell, it begins to accumulate as cell density increases, setting the stage for activation of the RhlR quorum sensing system. And so at low cell densities, the environmental concentration of our C4 homoserine lactone remains relatively low. However, as cell density increases, so does the accumulation of our autoinducer molecule. Now, at high cell densities, the autoinducer will have accumulated in the external environment, and due to its composition, just as it diffused freely out of the cell, it can diffuse freely back into it. And this is where our cytoplasmic concentrations start to increase. Now, whilst all of this is going on, the transcriptional regulator for this quorum sensing system, which is RhlR, has been transcribed and translated. And once a minimum activation threshold is met, our C4 homoserine lactone autoinducer will begin to interact with the RhlR protein. And this interaction triggers RhlR activation. This is where there's a conformational rearrangement of the RhlR protein, forming an active complex that is able to directly interact with specific DNA sequences, allowing it to turn certain genes on or off. Now, one of its primary targets is the RhlI promoter, leading to increased transcription of RhlI, further amplifying the production of the C4 homoserine lactone autoinducer via a positive feedback loop. And what this does is it ensures that once a quorum of bacterial cells is reached, the response is rapidly intensified. Now, beyond regulating its own system, the activated RhlR protein also controls the expression of multiple virulence-associated genes and biofilm-associated components. And finally, unlike the LasR system, the RhlR system only has hierarchical control over the PqsR quorum sensing system, and this is in a negative regulatory role. In other words, the activated RhlR protein downregulates components of the PqsR system. And essentially, this regulatory control is crucial because the PqsR quorum sensing system is a metabolically intensive system, and so by suppressing excessive activation, RhlR helps fine-tune virulence gene production, preventing unnecessary energy expenditure whilst maintaining a balance in the system. And so this naturally brings us to the final quorum sensing system of Pseudomonas aeruginosa, the PqsR system, which, as we can see, differs significantly from the LasR and RhlR systems. Now, importantly, it serves as a regulatory link between the two, and as we discussed earlier, activated LasR upregulates the PqsR system by inducing expression of the PqsR gene, which encodes for the transcriptional regulator for the PqsR system. And so once this has been transcribed and translated, it binds to the promoter sequence of the PqsR ABCD operon and activates its transcription. And what we end up with is a basal level of transcription for our PqsR ABCD genes. Now these are translated into their own proteins, but collectively, along with some other proteins, they form a biosynthetic pathway that allows the production of the PQS autoinducer. Now this autoinducer is a modified alkyl quinolone with the formula 2-heptal 3-hydroxyl 4-quinolone. Now this Pseudomonas quinolone signal, which is where it gets its abbreviation from, has a quinolone core featuring a double ring structure and a heptal hydrocarbon tail. Now, unlike the autoinducers of our LasR and RhlR systems, the PQS molecule does not freely diffuse across the cell membrane. Instead, its movement into and out of the cell is regulated by various cellular active transport systems. And these need to utilize a proton motive force to move materials across membranes, making it an energy-intensive process to move the autoinducer around. Now, this isn't great. However, it does allow for a more controlled accumulation of autoinducers within the extracellular environment. And so at relatively low cell densities, the environmental concentration of our PQS autoinducer remains relatively low. However, as cell density increases, so does the accumulation of our autoinducer molecule. Then, at relatively high cell densities, the autoinducer will have accumulated in the external environment, and at this point, the PQS signal molecules will accumulate within the cytoplasm due to either the PQS signal being imported into the cytoplasm or the export of the PQS signal slows down. Now, regardless of the mechanism, we get an increased cytoplasmic concentration of our PQS autoinducer. Now, whilst this is happening, our transcriptional regulator for this quorum sensing system, which is PqsR, is continually being produced. And once a minimum threshold has been met, our PQS autoinducer will begin to interact with PqsR. And this interaction triggers PqsR activation. This is where there's a conformational rearrangement of the PqsR protein, forming an active complex that is able to directly interact with specific DNA sequences, allowing it to turn certain genes on or off. Now, as we've seen previously, one of its primary targets is the promoter of the PQS ABCD operon, leading to increased transcription and translation of proteins in the PQS biosynthetic pathway, further amplifying the production of the PQS autoinducer via a positive feedback loop. And again, what this does is it ensures that once a quorum of bacterial cells is reached, the response is rapidly intensified. Now, beyond regulating its own system, the activated PqsR protein also controls the expression of multiple virulence-associated genes and biofilm-associated components. And I guess just like the last system, again, the PQS system has hierarchical control over the RhlR quorum sensing system, and this is in a positive regulatory role. In other words, the activated PqsR protein upregulates components of the RhlR quorum sensing system. Now, this might seem counterintuitive because if we remember, the RhlR system will downregulate the PQS system. However, together they allow Pseudomonas aeruginosa to fine-tune their quorum sensing response. Okay, so in this final section of the video, we'll summarize some of the global effects on a basic level. When cell density is high, the environmental concentration of autoinducers also increases. This leads to global changes in gene expression, resulting in the up or down regulation of specific genes within the bacterial cell. And so if we look at Pseudomonas aeruginosa under high cell density conditions, we see an increase in the production of several key virulence factors. The first is protease and elastase, which degrade host tissue, disrupt the extracellular matrix, and facilitate bacterial invasion. In addition to this, we see an increase in pyocyanin production. This is a redox active toxin that generates reactive oxygen species, leading to oxidative stress and host cell damage. There is also an increase in the production of cytotoxic lectins such as LecA and LecB, and these interfere with host immune cell function by binding glycoproteins on host surfaces. We also get the upregulation of siderophore production, including things like pyocyanin, which are critical for iron acquisition, and they allow Pseudomonas aeruginosa to compete for essential nutrients within the host environment essential during infection. And finally, one of the more crucial quorum sensing driven changes is biofilm formation, where bacteria transition from a planktonic lifestyle to a structured protective community that enhances antibiotic tolerance and immune evasion. Now, whilst all of this is going on, Pseudomonas aeruginosa will downregulate several key processes associated with early stage infection and motility. The first being swarming motility, a form of surface associated movement that is inhibited as bacteria switch to a surface associated lifestyle. Next is the downregulation of various metabolic pathways, shifting energy usage towards virulence factor production and biofilm maintenance. And finally, we see the suppression of the type 3 secretion system, and whilst this is crucial for acute infections, its downregulation suggests a transition towards a chronic infection state, where biofilm formation and the secretion of extracellular virulence factors play a more dominant role. And so ultimately, the LasR, RhlR and PqsR quorum sensing systems drive a major regulatory shift, promoting the upregulation of virulence factors essential for tissue invasion, immune evasion, and host damage, while simultaneously downregulating motility genes and those associated with a planktonic lifestyle. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.